Hello everyone, my name is Lloyd Cayman from Roxbury Community College. I am a science major and our honors class is going to be following up finishing work towards a project done by the Site 281 Research Science course. This project is a novel binding site on the COX-2 enzyme for aspirin. We will be talking a little bit about the famous drug targeting pain relief. First are some of the highlights of how our students work with chimera and Swiss stock to learn how small molecules interact with proteins, focusing on how to understand the COX enzyme and how it is inhibited by aspirin. We started by identifying the amino acids around the aspirin performing molecular docking after we did a review with assays for COX-2 activity and inhibition because we want to measure the activity of the COX-2 protein. Our goal of the project was to verify that we have found a possible new docking site for aspirin and other painkillers that are adjacent to the active site where it is known that aspirin participates in stimulation of serine 535 in the active site. This function has been known for some time in literature. Our goal of our project is to show that our inhibitors are inhibiting and may be docking adjacent to the actual site before diffusing it. This study can help with other drug designs for enzymes in this family, giving great interest in pursuing of results. To continue, we have identified amino acids in the novel binding site. We did it both with flexible and rigid docking, keeping careful, progressive notes for future research protocols for training our students. Now we are currently doing enzyme studies to prove that these mutants have new binding sites for aspirin and COX-2. Here we have some overlays before and after flexible docking. This is a little bit confusing, but the programs that we are using are all available online. The primary program students are using at RCC is University of California, San Francisco Chimera. With this software we can visualize the enzymes that we are working with and the small molecules when they are docked in the active site. Here you can see aspirin or acetyl and CH3 group double bond OO, and that group is a carboxylic acid, which is a drug made by Bayer in the 1890s, replacing a natural product called salicylic acid that American Indians knew about for centuries. Deriving from the willow tree, this is of a drugs classified as NSAIDs, non-steroidal anti-inflammatory agents. COX-2 is part of a biochemical pathway to make prostaglandins, so the phospholipids is the class of lipids whose molecule has a hydrophilic containing phosphate. These prostaglandins are part of this group. We have arachidonic, which is the long-chain unsaturated fatty acid, and unsaturated fatty acid, which lead us to postulate. Example, as we start with the phospholipid, we chop up the phospholipid enzymatically with enzymes, which is the chemical work. Then we make arachidonic acid, and then the arachidonic acid is converted to prostaglandin 2 by these selective COX-2 enzymes. As named, these are the inhibitors that we are trying to manipulate. They are known and commercially available if further study is of interest. But here what we are doing is learning new techniques to refine identification of novel binding sites for drug studies using known targets and studying these targets to identify new initial docking sites that accept the secretion of enzymes, already having known targets with our bioinformatics approach to drug discovery. This explaining the biology, the COX-2 and enzymes are controlled points that I just talked about. This leads the conversion of arachidonic acid to prostaglandin E2, causing pain and inflammation. Aspirin is used to inhibit or block this effect. Here you are inhibiting the COX-2 enzyme with the aspirin to improve where that binding site is. We have to inhibit the inhibition so we get activity. This is the full pathway, in which here you can take a look. Giving background of the function we have studied, there are two COX enzymes constitutive, meaning that COX-1 enzyme is on all the time in the stomach, kidneys, and platelets. The one enzyme that we are trying to target always is the COX-2, which is why you know some children can't take aspirin because they get perspectivity or issues arise, so we're always looking for more specific inhibitors of COX-2. There are many known, but there is still lots of work to do in this area. To finish, here are what the enzymes are doing to promote homeostasis in the human body. Their functional role sometimes gets out of whack in its pathophysiology, which pertains to an unhealthy role of function. When this happens, we have to control the inflammation with the use of NSAIDs, which are these type of over-the-counter painkillers. In the first part of the study, all of us as students were looking at the binding pocket of the enzyme with the Chimera software. We used the camera software to visualize the protein. Here we got the COX-2 coordinates out of the protein databank and inserted them into Chimera to be studied and map out where the center was based on the secretion of salicylic acid, which is the natural inhibitor. Then we docked the aspirin, where we started to see the differences in the way the aspirin would bind when the crystal structure had the salicylic natural inhibitor secretion. But when we put the aspirin in, we saw some differences in the way binding occurred, so we identified some key residues that we believe aspirin is binding with deeper entry into the active site's cavity. These residues are listed here. The bold is what happened when we mutated those residents from arginine to alanine, preventing hydrogen bonding, and we got no inhibition, which means we knocked out the inhibition of aspirin when we mutated. When the serine was mutated to alanine and other amino acid, we also got no inhibition. When we say we got no inhibition, that means aspirin no longer bound. So aspirin could not inhibit COX-2, which means aspirin is probably binding to these residues, because to inhibit COX-2, aspirin would have to bind to these two. Without having alanine present, A stands for alanine, which is a mutant, a different amino acid, we lose the effect of inhibition. Now if you see inhibition when this residue threonine 118A is mutated to alanine, that means it is not essential for binding the aspirin, but you see a lot of these are like valine 116A to alanine having no inhibition when mutated. Which means aspirin could not bind because aspirin is the inhibitor. Here is another picture showing the flexible docking again. And now we see part 2 with the flexible docking 116A, 119A, and 120A again, because it showed up in both flexible docking when we allowed the side chains of the enzyme to move and in rigid docking when we did not. But there were two other enzymes that showed up with a flexible docking when we allowed the side chains to move, and that was tyrosine and threonine. Now, having a complete view of the goal and project, we understand where the binding site are physically in the three-dimensional projection of the protein. By having the coordinates of the protein from the Rutgers Consortium of Structural Biology, we did the docking by Swiss dog. From here we now know all the amino acids that we think from mutations that were done in our lab. Next is to conduct full kinetic studies on them, doing different concentrations of the inhibitor to get the binding affinities. We're still working on that. Principles of Chemistry 2 is supposed to finish up all those binding constants. So this is an overview of how we did the work, and now we are looking at other inhibitors that we know work as controls to make sure our assays are working. To know we are not working with silly COX NSAIDs, it is just one of the inhibitors we got. I have an explanation. If you have more questions, you can contact us. Furthermore, our mutagenesis is a technique that is well known. It is a site-directed mutagenesis. Mutagenesis, a process involving the recombinant of protein. For those of you not familiar with that, this is the cloning of the COX-2 enzyme, human COX-2 enzyme, running in a plasmid where they are overexpressed with the black operin and purified. That protein and function of the mutagenesis is to define the binding pargament. Our mutations were done last year inside 281, where we had different primers for each meeting, and we expressed the protein and froze it down for the Principles of Chemistry students to use in these honors projects. Here you can see what happens. These are different types of mutations, called insertions. An example of an insertion is if you have ADN as the original, you change one amino acid, in this case from T or threonine to cysteine. We did it here from the list that I showed all of them to alanine. As a result, they could not sustain hydrogen bonding. We also have used our assay kits to validate our inhibition. You can see the details of the kits that we use from Abcam with celecoxib as a control, and we'll be showing that data in future talks. Now that we have confirmed that we have overexpressed the protein and that we've lost some activity with the mutants, we conducted a full kinetic curves on them. This is how the fluorometric assay works. Furthermore, we had a great deal of questions that we had to be able to answer to do this project. We needed the positive controls, which are the celecoxib and the Celebrex and the aspirin, which themselves are known inhibitors of COX-2 for reference whenever we worked with the new assay. From here we designed a novel foreseen assay. We also worked with assays commercially and changed them or adopted them for 24 well plates and different applications. But we always had to understand the positive control. It allowed us to know what we were detecting and observing in the experiments. Knowing that the positive control was working gave an idea of what we were predicting and what we should see when the inhibitors were added to the symbols. These are the different answers that we learned from our work. Thank you very much for your time, and let us know if you have any questions.